Hi everyone, welcome to my channel Pure Biology, and today I come with the continuing video of the functions of the endoplasmic reticulum, and today I will try to explain about the formation of the disulfide bond that is occurring in the endoplasmic reticulum, and obviously this is in RER, okay, RER, and this is obviously in the ACR, okay. So, this is the actual topic, the formation of the disulfide bond. This disulfide bond, do not form in the cytosol due to the reducing environment which maintains cysteine residues in their reduced state. Okay. And this bond formation in endoplasmic reticulum lumen is facilitated by the protein disulfide isomers. Okay. So, what is saying that this disulfide bond do not form in cytosol or cytoplasm. Why? Due to reducing environment, and this reducing environment maintains cysteine residues in their reduced form. Okay. So, in the cytoplasm, the cysteine residues that remain in the reduced form, and that's why the environment is the reducing environment. So, that's why this disulfide bonds, that means this type of bond, these do not form in the cytoplasm. So, where it is formed? It is formed in the lumen of the endoplasmic reticulum because of the presence of, you can say, the enzyme that is called the protein disulfide isomerase. This enzyme present in the endoplasmic reticulum lumen, that's why here the disulfide bond is formed easily. It also catalyzes rearrangement of the disulfide bond. The enzymes play another role in the rearrangement of the disulfide bond. That means if there is any mistake is in the formation of disulfide bond, so this mistake is corrected by this enzyme. So that's why it is said that it also helps in the rearrangement of this disulfide bond. Generally, in the bacterial cell, disulfide bonds are found in the periplasmic space. Okay, periplasmic space. This is the place. Obviously, in the bacterial cell, you can see this space. Okay, so when I discuss about the bacteria, then I will try to explain about this space. And now, these disulfide bonds are found only in the secretory proteins and in cytoplasmic domains of membrane proteins. Cytosolic proteins and organelle proteins synthesized on free ribosomes lack the disulfide bonds. Clear? So, where you can see these disulfide bonds, generally these are found only some secretory proteins. Okay, some secretory proteins, this is obviously seen, and in cytoplasm domains, okay, domains of the membrane proteins, generally in the proteins. You just memorize that in the proteins this type of bond is seen or found. And the proteins, exceptional case obviously, some cytosolic proteins and organelle proteins that are synthesized on the free ribosomes, they lack the disulfide bonds. Okay, so in only you can see this disulfide bond in the bacterial cell. Bacterial cell means the bacterial proteins, you can say. Okay. So this is the we know that the formation of the disulfide bonds, this is possible in the endoplasmic reticulum lumen, and due to the presence of one enzyme, that is the protein disulfide isomerase. That's all. Next is the detoxification of xenobiotic compounds. This is the another role of the endoplasmic reticulum, but this is actually the role of, you can say, ACR, or the smooth endoplasmic reticulum. Generally, smooth endoplasmic reticulums in the hepatic cells are the major site for removal of xenobiotics, cytochrome. So this is the actual space or place where in the hepatic cells, okay, the ACRs that present in the hepatic cells, these are the major site for removal of xenobiotics. Okay. Next, what is the role of the cytochrome P450? This cytochrome P450 that present in the ACR, okay, smooth endoplasmic reticulum. So this cytochrome P450 that present in the ACR, this catalyzes this process. Okay. So which enzyme do you have to memorize that the name of the enzyme, that is the cytochrome P450. With the help of cytochrome P450, the removal of xenobiotics is possible. Where? In the ACR. Clear? This cytochrome P450, these enzymes are the hemoproteins. Okay? This is the hemoproteins and monooxygenases catalyze the incorporation of a single atom of molecular oxygen into a substrate with concomitant reduction of other atom to water. So here the cytochrome P450 is that hemoprotein. Okay. Okay, so here this cytochrome P450, it is one type of hemoproteins, and the enzyme is the monooxygenases. Okay, this is the actual mixed function of oxidases, and these monooxygenases, this catalyze the incorporation, or you can say that inserting, or it helps in attachment, you can say. The incorporation of a single atom of molecular oxygen into a substrate with the concomitant reduction, or you can say the reduction of other atom to water. So it helps to insert single atom of molecular oxygen into a substrate by the process of reduction of another atom to water. That means it helps to reduce other atom into water, and by this process it tries to insert oxygen molecule into a substrate. Clear? They are ubiquitously distributed and multi-catalyst. Okay, these are ubiquitously distributed multi-catalyst. And cytochromes P450, why this name is given? Because they got their name from their character. What character? As the hemoprotein, and from their unusual spectral properties that displaying a typical absorption of maximum reduced CO bound complex at 450 nanometer. Okay, so why this name is given? Because first of all, they are hemoprotein and they have the capability of absorption the maximum of reduced CO bound complex. They can absorb maximum of reduced CO bound complex at 450 nanometer wavelength. Clear? That's why P450 name is given. Generally, they use the molecular oxygen for what? To catalyze any reaction like that hydroxylation, sulfoxidation, deamination, etc. And they get electrons for this continuation of these reactions. They get the electrons from the from the NADH or NADPH. Okay. That means they get electrons that is necessary for the oxygen activation or substrate hydroxylation from where? From NADH or NADPH. Generally, the action of the electron transferring proteins. So, via the action of the electron transferring proteins, in that way they get the electrons. Okay, without electrons, oxygen activation or any substrate hydroxylation is not possible, because electron transporting is necessary here, and by for this continuing the reaction or for this completion of the reaction, they get the electrons either from NADH or NADPH or any electron transferring proteins. Clear? So these are the actually the formation of disulfide bonds in RER and detoxification of xenobiotic compound. These two functions that is occurring in the endoplasmic reticulum I have explained here. Okay. So in the next video I will try to another function of the endoplasmic reticulum, that is the synthesis of the membrane lipid. Okay, synthesis of the membrane lipid. So continue with me and my channel. Thank you.